Hello friends, today I am going to present a new video on microbes in human welfare. This is the chapter number 10 of NCRT. So let's start. In this chapter, we'll study the topics microbes, microbes in household products, microbes in industrial products, microbes in sewage treatment, microbes in production of biogas, microbes as biocontroller agent, microbes as biofertilizers. So let's start this chapter. So our first topic is microbes. So what is microbes? Microbes are macroscopic organisms which are invisible to naked eyes. They are present everywhere on the earth, in soil, water, air, in our bodies, plants, and even at the most extreme locations such as hot water geysers and poles. Microbes are diverse. They can be protozoa, bacteria, fungi, viruses, viroids, pyrons, and etc. Microbes are thought to be the main disease causing agents, but they can be helpful to human beings in many ways. So let's study the examples of microbes. So the first example is E. coli, which full form is Escherichia coli. Second is Lactobacillus. Third is Methanobacterium. And fourth is Azotobacter. So our next topic is microbes on household products. In this, we will study the production of curd from milk. Microbes such as Lactobacillus and group called lactic acid bacteria converts milk to curd by producing acids which are partially digest and coagulate milk proteins. A small amount of curd, which also known as inoculum, is added to milk contain millions of bacteria which multiply under a suitable condition and thus converting it into the curd. This process increases the nutritional value of milk by increasing vitamin B12. It also checks diseases causing organisms in our stomach. The next topic we will study is fermentation. The fermentation of dough is due to the bacteria and its puffed appearance is due to the production of carbon dioxide during fermentation. 2D, an alcoholic drink, is made from fermentation of palm sap. A yeast, which scientific name is Saccharomyces cerevisiae and also known as baker's yeast, is used for fermenting bread dough. Cheese is also a product of fermentation, and different varieties of cheese are due to different microbes used for fermenting. The holes in cheese are due to the large production of carbon dioxide. So, in this diagram, you can see in the Saccharomyces cerevisiae as seen under the DIC microscope, which full form is differential interference contrast microscope. And the second picture, you can see the Swiss cheese. The large holes in Swiss cheese are due to the huge amount of carbon dioxide released during fermentation by Propionibacterium shermani. So our next topic we will study is microbes in industrial products. In this, we will study the beverages, antibiotics, organic acids, enzymes, and bioactive molecules. So first topic we will study is beverage. In this, when juices and malted cereals are fermented, they get converted into ethanol. Brewer's yeast, which scientific name is Saccharomyces cerevisiae, is commonly used for fermentation. Drinks with versatile color and flavor are produced depending upon the raw material and nature. Classification of beverage. Beverage is classified into distilled beverage and non-distilled beverage. In distilled beverage, there is whiskey, rum, and brandy, and in non-distilled beverage, there is wine and beer. Now, the second we will study the antibiotics. The term antibiotic means against life with reference to disease causing organism. The first antibiotic is penicillin, which was discovered by Alexander Fleming. Penicillin is obtained from a fungus Penicillium notatum. These are used in treatment of number of diseases such as diphtheria, leprosy, whooping cough, plague. Our third topic is organic acid. Various organic acids are produced with the help of microbes. The below list of the organic acids and the name of microbe employed in producing it are given. Organic acids, first is citric acid, whose microbe is Aspergillus niger, which is a fungus. Second is acetic acid, whose microbe is Acetobacter aceti, which is a bacteria. Third, our organic acid is butyric acid, whose microbe is Clostridium botulicum, which is also a bacterium. Fourth is lactic acid, whose microbe is Lactobacillus, which is a bacterium. And fifth and last is ethanol, whose microbe is Saccharomyces cerevisiae, which is a yeast. Now, fourth, we'll study the enzymes. So, first enzyme is lipase, which is lipid dissolving produces by Conidia lipolytica. Second enzyme is proteases. Third enzyme is pectinase. So, lipase are added in detergent for removing oily stains. Pectinase and proteases are used to clear the fruit juices during the bottling. Streptokinase is produced by Streptococcus. It is modified by genetic engineering and used as a clot buster for dissolving blood clots of patients after myocardial infarction. Our fifth topic is bioactive molecules. In this, we will study the cyclosporin A and statins. So, in cyclosporin A, the fungus which produces it is Trichoderma polysporum. It has immunosuppressive properties, so it is used as immunosuppressive agent during the organ transplantation. In statins, there are it is the product of fermentation activity of yeast. The yeast which produces it is Monosaccharus purpurus. It inhibits the cholesterol synthesis, so used for lowering blood cholesterol level. So, as you can see in this diagram, there are antibiotics and there are bio, a product of fermentation, acetic acid, the main component of vinegar, cholesterol lowering tablets, statins. So our next topic is microbe in sewage treatment. Why we need sewage treatment? The municipal waste water contain large amount of organic waste and microbes. This can invite disease and epidemic when this water meets community water bodies like lake, pond and rivers. To check this sewage is made less polluting by passing it through sewage treatment plant. Short form is STPs. This involves two steps. First, primary treatment and second is secondary treatment. So first we will study the primary treatment. In this, it is a physical process. It uses the processes sedimentation and filtration to filter small and large particles. First, the sewage is passed through mesh screens of the small pore sizes to remove floating objects like polythene. The grit is removed by sedimentation by passing sewage into grit chambers. Then it is kept in settling tank where the suspended material settle down to form the primary sludge. The remaining fluid is taken for secondary treatment. So in secondary treatment, it is a biological process by heterotrophic bacteria present in the sewage. The primary effluent is aerated in the aeration tank. This causes the rapid growth of aerobic bacteria, which then act as a flux, consume the organism matter to reduce the biochemical oxygen demand. After this, the effluent is passed to the settling tank where these flocks get sedimented. A part of this sludge is pumped back to the aeration tank for further use. This sludge is pumped to anaerobic sludge digester where the anaerobic bacteria form gases like methane, carbon dioxide and H2S. After this, the effluent is released into the water bodies like streams and rivers. So as you can see in this diagram, microorganism in sewage treatment. So our next topic is microbes in biogas production. So biogas consists of methane 60% and carbon dioxide 40%. It is produced by methanobacterium by acting on cellulosic compounds. These are found in rumen of cattle and anaerobic sludge of sewage. Cattle dung contain both cellulosic as well as methanogens, so it is used for biogas production. It is a popular method for meeting energy requirements of rural household and also help in reducing the precious firewood. Second, the steps involved in the production of biogas. The biogas consists of a 10 to 15 feet deep concrete tank in which the slurry of dung is fed. A floating cover is capped over the slurry which keeps on rising as the gas is produced in the tank by microbial activity. An outlet connected to a pipe is present in the biogas plant for the removal of biogas and supplying it to required place. The spent slurry is removed through another outlet which can be used as fertilizer. This is the diagram of typical biogas plant. And this is the biogas plant in Tamil Nadu. So our last topic is microbes as biocontrol agent. So biological control is defined as a reduction of pest population by natural enemies and typically involves an active human role. For example, the spores of Bacillus thuringiensis, short form is BT, are toxic to certain larvae but not harmful for others. The fungus Trichoderma is being used for fighting plant diseases. Baculoviruses are the pathogen used for attacking the harmful insects and arthropods. They belong to genus Nucleopolyhedrovirus. The use of biofertilizers instead of chemical ones has been known to keep the soil fertile intact for longer duration. They are the organisms like bacteria, fungi, and cyanobacterium that enrich the soil. A. Bacteria. Rhizobium azopyrillum azobacterium fixation atmospheric nitrogen and enrich the nitrogen content of the soil. Fungi, they form the symbiotic relation with the roots of plant, example mycorrhiza. It absorbs phosphorus and passes it to the plant, and also it shows the resistance to the pathogen and salinity. C. Cyanobacterium. They are the autotrophic microbes which can fix atmospheric nitrogen. For example, Anabena, Nostoc, Oscillatoria. They are important as biofertilizer in paddy fields. So here we ended with this chapter. I hope you all like this video.